So I would like, first of all, of course, to thank the organizing committee for giving me a chance to come here and make this talk in front of you. It's really my pleasure. I will speak today about an interplay between microRNAs, transcription factors, and target genes. It's a recent publication from this year. In fact, long story short, this is the presentation and the paper. That's it, if you can get something out of it. Mostly we started looking at microRNAs. MicroRNAs, as you well know, are small non-coding RNAs, about 22 nucleotides, which mostly negative post-transcription regulate mRNA level. Kind of canonical microRNA production scheme is presented here. It's canonical, it's simplified, but well accepted. Not always true, but still. MicroRNAs are produced in nucleus, then they are cleaved from pre-MER by Drosha complex. The resulting pre-MER is exported, a double-stranded pre-MER exported outside, where it's cleaved by dicer and afterwards is incorporated into RIS complex, one of those strains. The second strain is historically called a passenger strain, and it was thought to have no effect, to be completely degraded. However, the recent work suggests that the second a passenger strain can have at least the same level of effect. One of papers where it was shown, this paper from our group 1 year ago. It's known that microRNAs are involved in quite a lot, perhaps all cellular processes and diseases. About 60% of genes are predicted to be targets of microRNA. The estimation is about, let's say, 60% for the moment. And the role in cancer is shown, well described. For example, some microRNAs work as tumors suppressing ones like miR15A, some working as oncogenes, miR21. That's an overview of one of those two targets which we took into account in our work. The problems with microRNAs is that they are short and in principle their sequence is not necessarily should be complementary 100% to a target gene, which hampers somehow the prediction of microRNA binding and targets. We have checked for quite some databases. In fact, if we overlap 4 of them, what we get after a prediction in Venn diagram in the center was 0, right? So it's still, there is no concordance, so we have to take into account data and experiments, especially because microRNA expression depends really on cell types and biological conditions. And the precise mechanism of microRNA action are still debated. To understand those mechanisms, in fact, to solve reverse engineering tasks, it's not enough to look on the cross-sectional studies where we have a problem with defining what is a cause of regulation and what is a response. We have to go for a dynamic regulation. We have to take into account the time series to answer this question. The goal of our work is aiming on a time series, try to integrate microRNA data, mRNA data with existing knowledge about their functions and targets in order to get some new information, hopefully, about interaction after stimulus in a cell line. Here we found a robust list of microRNA target genes, so microRNAs which downregulate mRNAs, compose and verify pipeline for analysis of time-resolved microarray data, finding quite strange results, but seems to be proved. Investigate dynamics of active biological functions, how functions behave in time, let's say. Integrate microRNA, mRNA, and upstream regulators, which we inferred in a global view, and try to detect, for the moment by eye, some topological motifs in the regulation network. The feed-forward loops, for example. This is experimental perturbation which we made to our system. This is a standard, well-known interferon gamma activation of JAK-STAT signaling pathway. Interferon gamma activates JAK1 and JAK2, which then results in activation and making a complex of STAT1 transcription factors. The complex is transposed into nucleus and activates many other pathways, including transcription factors, genes, and so on. So this is well known. What we added as additional control here is inhibitor, JAK inhibitor 1, which targets and stop activity of those two kinases, which results in a kind of blocking all this downstream pipeline. It was an additional control. What we've done, we seeded the cells at A375 melanoma cell line cells were seeded in triplicates in the same time point, in the same moment, let's say, and then they are growing for 96 hours, in fact. Then at some specific time points, we add a single stimulation by interferon gamma, so it was just one stimulation, and see what happens with the cells after this. All cells are collected at the same time point, including the one with JAK inhibitor, an additional control, and the mRNAs, or let's say RNAs extracted, was put into, analyzed by two different slides, Affymetrix human genome 1.0 ST arrays and Affymetrix microRNA version 2 arrays. The nice point about this kind of experiment is that we have from the same plate, let's say, the same well, we have mRNA-OM, messenger RNA, and microRNA coming from the same well. The analysis of the microarrays was performed in a standard pipeline. We use Particle Genomic Suite, but of course one can use Affymetrix Power Tools. GCRMA correction was used for mRNA cell files. We use gene summarization to get gene expression from prop sets. For microRNA, we use RMA just and filter features which are specific only for human, and we took into account only mature. Unfortunately, immature, or maybe fortunately, had very low level of expression, so we keep only the mature one. The only difference is that we put here additional filtering step, where we removed all lowly expressed features. And this really helps us somehow to improve number of differentially expressed genes. We've checked this, performed the analysis with different threshold, where we filtered out the lowly expressed genes, and you can see from these normalized Z-scores of number of differentially expressed genes, there are an optimum. Fortunately, or in our case, let's say, it was at the same position with a threshold of 7, log 2. This is an overview, let's say a phase space, a kind of phase space of what happened in those cells at general transcriptome level, mRNA and microRNA. How we generated this plot is that we simply took the principal components after analysis of mRNA series, after analysis of microRNA series, and those first principal components are shown here with X and Y, microRNA and mRNA. Each point is an experiment. So what you see in the beginning, without big changes in microRNA level, mRNA level modifies quite a lot. Then it's more or less with the same strengths, microRNA, mRNA, and in the later time points, mRNA-OM, let's say, does not respond, while in microRNAs we still have some differences. So directly from here, we got the idea that perhaps something is happening with a delay in response. The pipeline which we used was rather standardized, rather standard one. We used the co-expression analysis to detect potential microRNA targets. Then we used differential expression analysis to get significantly regulated MIR and mRNAs. which are afterwards used in Ingenuity pipeline to detect the functions activated. We inferred upstream regulators, taking into account those significant genes, and afterwards combined the transcript regulators functions, significant mRNA smear in an integrative way in order to generate a kind of integrative picture with circos and detect network motifs after. So let's start. The co-expression analysis was done by our own tool. It's a freely available, user-friendly tool for a quite simple one, I would say. The nice feature of it is that it's allowed not only to browse and to find the correlated profiles, but also to combine it with a known data which you can upload there. Interesting but expected observation which we got is that number of negatively correlated mere mRNA pairs. For example, for this mere, you can see that the blue one corresponds to negative correlations, the red one is for positive correlation. So negative correlations are really more oftenly seen between those. If you modify the threshold, number of negative correlations goes up always, which is something what would we expect from the regulation. Unfortunately, using only co-expression, we were not able to get anything interesting because if you look here, this is the density plot for the correlation coefficients. It's really hard to get a good false positives ratio here. So because we have only 4 independent time points, more or less, we have to combine the experimental observation with a data set. It was done, and we observed about 400 predicted microRNA mRNA interactions and 17 known published interactions coming from the tarbase. This was the first part of the story. Then we go for differential expression analysis. And doing this, you can ask yourself the question, 2 kinds of questions, which genes are changing and which genes are changing in each specific time point. And we tried several statistical packages to do this kind of analysis. Lemma, which you know doesn't take into account the time correlation, just consider time points, independent time points, but is well established. BATR takes into account the time correlation, and time course, specific package for the time course analysis. And those were tested on mRNA data, microRNA data, and permutated data in order to detect permutated FDR, and also synthetic data, where we know beforehand which genes are regulated, which are not. Interesting result which we get are that, strangely, Lemma outperforms those 2 methods in our data set, and we validated it with MeEV software tool from the developer of BATR as well. So with BATR and time course, we had problems with FDRs, in permutation FDRs, which are not the same as we expected, and the real FDRs calculated on simulated data set. With Lemma, everything is really in line, even almost the same estimation as was predicted. Those are results of differential expression analysis. We were able to define several clusters, gene expression and mirror expression. You can see that there are 3 genes expression going up and down, going down in the top 50. In mirror, it's mostly kind of binary, up or down. And interestingly, the number of differential expressed genes is really lagging in microRNAs. Mere responding after between 12 and 24 hours, let's say. At 24 hours, we have uprise, and for mRNA. For mere, we have uprise at 48 hours. So you can see from the profile, something is happening there. We've checked also what happens with the machinery of microRNA production, and interestingly, we found that dicer, the part of Drosha complexes DGCR8 and AGO2 were upregulated exactly at the same time points. It doesn't happen with RNA-seq for some reason, but still it's quite highly expressed. And this corresponds to this uprise time point. Next, we go for functional annotation. What you can get nicely from IPA or from another enrichment analysis is the heat map of the p-values. It's a log transformed p-values, which according to the colors here, you can identify early regulation, intermediate, let's say, regulation always presented there, and late regulations. This is what published. I will not speak too much about this. I will switch to this picture. This is something we do not put in the publication. It's the same, more or less, representation, but here we took into account not only p-value, but also a z-score, which is not available for all functions, unfortunately, but for some at least. And it's nice because what you see here is not only whether the function is significantly enriched, but whether the function is up-regulated or suppressed. We can see what happens in the beginning after putting interferon gamma. We have, where is it? Yeah, it's here. We have a cell death. The cell viability is also enriched. The death. What we have, anapoptosis also early. So the cells starting to die. Then we have a second part where we have an immune response for the cells and also the differentiation of cells. What I'm going to say that now is a kind of, it might be not true, but it's a speculation about this data. It seems that the cells feel really stressed in the beginning. They produce a response. And after what we see in the later time points, we have downregulation of quite some apoptosis-related functions. So cells are not dying anymore, but they're also not proliferating, which was observed phenotypically, by the way, and not moving. It's a kind of result. So cells are surviving, but in a kind of stationary state after this. Next, we go for the last part, an integrative analysis. First of all, Ingenuity provide us with the possibility to detect the master regulator, upstream regulators, which are transcription factors more often, or other transcription regulators. It can be also MIR, by the way, microRNAs. So it took significantly regulated genes and tried to find out which regulators can explain their differential expression. By this inference, we kind of put into our analysis protein level. Of course, it's an inference. It was, the results were visualized with the circus plots. We put differentially expressed genes in different categories. Here we have transcription regulators and several MIRs. We put just a few of them, those which are linked the most. And we can see what happens in time. It starts with regulation of genes, some transcription factors. For example, IRF1 is the next one after STAT1. So STAT1 activates IRF1 and it immediately have an effect. And then afterwards, this regulation increases. At 24 hours, we have most dense picture between the regulators and the genes. MIRs are not for a moment playing a role. Then MIR come to a play. We have a decrease of those regulations by transcription factors. And picture 72 is even, I think, less dense. For the control, IRF alpha is still there. For the control with inhibition, IRF alpha is still there. IRF1 is still there, but the regulation after 72 hours is very similar to an early time point. Nothing is changing much. We detected several motifs. Unfortunately, we were able for the moment to detect them only by eye. So we scanned through those Ingenuity pictures looking for the connection, the subnetworks which make sense. And there are a few of them here combined in one subnetwork. NF-kappa-B activating ICAM1, for example, and activating MIN21, which has a downregulation effect. By colors you see here whether we can see those lines, those connections, not only from our databases, from Ingenuity, but also from expression level or activation level. Quite some of them are really seen by eye if you look on the profiles. So I'm always there. To give a kind of summary, we observe a delay response of microRNA with respect to mRNA after interferon gamma stimulation. Again, a speculation, but it seems like microRNA are involved in a fine-tuning and compensation of what happens in the cell. We detect some promising regulating motifs and validate a pipeline for simultaneous analysis of time-resolved microRNA and mRNA data. Showed the benefits, at least in our data set, of the standard pipeline lemma, and that the filtering of lowly expressed genes can really improve number of differentially expressed, which is more or less obvious. Thank you very much for your attention. Just the very last slide, I would like to mention, of course, my first co-author on this paper, our PhD student, Susan Reinsbach, who is a biologist and have done all the biological experiments. Bioinformatician Arno Müller, who worked with Ingenuity, Natalie Nicot, who produced nice microarray slides, Dimetra doing the qPCR analysis, my boss, Laurent Ballard, and the initiator of this project, Stefan Kreis. Thank you very much.
Okay, thank you very much. We are fine in time, so we have time for a couple of questions. Other questions?
You mentioned that you used a tool to infer the master regulators, the transcription factors, and the microRNAs. You didn't describe the specific method of how you went about inferring those master regulators, so how was that actually done?
Yeah, the good question. Ingenuity, specific statistical methods for Ingenuity, it seems for me that it was a Fisher test again. So we have the master regulator, we know all those, we have the transcription factors, they have an idea which genes it regulates, so a kind of category. Is this category enriched or not enriched? If it's enriched, then it's working. The second point, if it's enriched and let's say in the positive sense, the genes are upregulated, then this transcription factor having positive effect will be scored with positive Z-score. If it has a negative effect, it should be scored negatively then, more or less like this. But unfortunately, Ingenuity is not open tool, so always when you work with these business tools, there are kind of things that's in itself a black box.
This makes me think of the keynote talk from this morning. I think that has some relevance here. But I guess so, okay, so you basically were looking in databases of known transcription factor gene relationships.
Yes.
Or the tool was doing that, we think, but we're not really sure since it's not open and apparently not disclosed. So that's probably then far from complete.
Yes,
there are a lot
definitely.
of unknown relationships.
For me, it would be really nice if you could get something open, something which we understand from the beginning to the end, including the methods used. But for this specific paper, we decided to keep a standard pipeline.
Okay.
Standardized pipeline.
I don't know if our keynote speaker from this morning is here, but I think she would strongly encourage you to make that happen.
Thank you.
Thank you.
Are there more questions? So I have one. So you mentioned that you found like hundreds of new microRNA targets and a couple of them already in TargetDB. So how many of these could you actually validate by other means?
In fact, I don't remember precise number, but I think 20 we validated with qPCR of 23. So 23 we checked, 20 we were able to validate with qPCR. But of course, this validation speaks about co-regulation. We have to do something to silence it, to disturb another way. to be sure that it's really a direct connection.
Was the fraction higher for the new ones or lower as compared to the target TB ones?
I would say that we haven't checked this, first of all, but those connections which we found are in fact those which have the prognostic, which are found also in those databases. We have filtered out for the moment everything which is just co-regulated. Just because the false positive rate was so high, we were not able to keep only the data-driven approach.
Thank you. So, I do not see any more questions to this talk. So, thanks again. And we start with next proceedings presentations in 3 minutes or so. Thank you.